So we left off talking about biofilms, and one of the important aspects of biofilms is that the cells within it can communicate. One of the ways that bacteria communicate is through quorum sensing. And quorum sensing means when there's enough, the bacteria start to influence gene expression from other bacteria. So you see low bacterial concentration, they're producing things, there's not much reaction going on. But at a higher bacterial concentration, now the cells are signaling or communicating or telling each other, hey, express this, express that. And that's how you get this nice buildup of this biofilm layer and this protective coating that kind of keeps everybody in this community safe. And like I mentioned before, the issue is it's hard to get drugs in through the biofilm in order to kill off the microbes there. So quorum sensing is a method of bacterial communication based on bacterial concentration. Another feature that some bacteria can do or produce to make themselves more resistant for the environment is to produce endospores. So endospores are formed when the environmental conditions are unfavorable. So maybe there's low, I can't really talk and write at the same time, low nutrient supplies. And so the bacteria decides, I'm not going to survive very well. I need to make this little endospore and basically save my genetic information. And there's a nice coating around here that protects it from drying out. It's kind of like a hibernation. It's a dormant state that these endospores can last for years. The two main endospore producing bacterium are Clostridium, I'm not writing very well, and Bacillus. So in a lot of nursing homes and hospitals, you might have heard of C. diff. That's an abbreviation for a type of Clostridium that is produced, especially when people get sick, a weakened immune system, and then the spores can be released and it can infect other people. And so a lot of times people with a C. diff infection are actually quarantined from everybody because it can so easily spread through these spores. A couple other things to understand about bacteria is that we talked about cell division is basically making clones or identical copies of the bacterial cell. Okay. Bacteria, to evolve, need to get new genetic information. So that can always happen by mutation, but mutation is slow, and mutation doesn't necessarily give you brand new characteristics. So bacteria can actually get new genes, new genetic information from outside sources. Okay. So one is called transformation. And in transformation, bacteria are actually getting DNA from the environment. So what happens in the environment is that bacteria die all the time, their DNA is released, and some bacteria can actually take up that DNA, and sometimes that DNA ends up integrating into the genome and hangs around and can give a whole new characteristic to the bacteria. Not a lot of bacteria can naturally do this. This is really important in biotechnology or molecular biology. We use transformation to force bacteria in the lab to take up new genetic information. Transduction over here is another method of gene transfer, and this is by phage. So remember, these are viruses that can infect bacterial cells. So sometimes the phage will actually grab DNA from a cell that it's infecting, and when it goes to infect a new cell, it transfers that DNA. So another way that bacteria can get a whole gene instead of waiting for mutations to slowly change things. Probably the most important way for gene transfer that we're concerned about in the medical field is conjugation. And we also like to call this bacteria sex. And what's happening in conjugation is numerous genes are delivered, transferred. Okay, and you can see this can be through a plasmid, or this can be chromosomal DNA. And you probably learned about this in genetics. Conjugation, we are finding that organisms we didn't think could mate or have sex are actually being able to conjugate, and this is a huge issue for antibiotic resistance and virulence factors like toxin production. So 3 main ways bacteria can get whole genes from the environment, from a different bacteria by means of phage transfer, or from direct contact with a bacteria through conjugation. Other features of bacteria that can change the genetic makeup are transposons. These are also called jumping genes. So they have sequences around them that can actually cut themselves out and put themselves into new DNA locations. So they might move to a different site in the chromosome, or they might move to a new plasmid. Again, if you look back here, then you can figure out how these jumping genes could be transferred to other bacteria. And this is just an example of a plasmid. Remember, plasmids are self-replicating. They're outside the chromosome, so we call them extrachromosomal. Okay. Here, all these green ones are different antibiotic resistance genes. Here's an operon that allows for, like we said, coordinated gene expression. So plasmids can start accumulating lots of different genes from lots of different sources. And this is just an example of a Shigella plasmid that we have found transferred to different organisms, and it's called a virulence plasmid because it's giving the organisms virulence genes, that make them more pathogenic. Oops, sorry, my other computer is beeping at me. Okay, a couple more slides. Gene expression, I mentioned back here, an operon, which I guess I haven't really defined yet. So an operon is a way that bacteria can regulate multiple genes to be expressed at the same time. So both of these allow coordinated gene expression. And what that means is, in response to the environment, bacteria have organized their genes so that they will turn everything on at once that's needed. say to metabolize a certain type of sugar, or to now produce flagella so that it can move. So the difference between an operon and a regulon is an operon has one promoter with multiple genes under that single promoter. They're all in a row, and they make what we call polycistronic RNA. Okay. So what's happening is the RNA that's made encodes a whole bunch of genes, and then from translation you get individual proteins. Okay, so one RNA, multiple proteins then translated. A regulon uses the same regulator protein, so like an activator, which binds to multiple promoters to turn on the genes. Right? So you might have one gene, you might have polycistronic genes, but the point is coordinated gene expression. Quick way to respond to the environment by turning on all the genes or turning off all the genes if you don't need them. Okay. Here's a little closer look at the phage, which are viruses that infect bacteria. We talked about the idea of transduction as a way for bacteria to obtain new genetic information, and that's due to the movement of these little viruses. So again, hopefully you remember from micro or genetics that some phage go through a lytic cycle, which means they infect, they make more virus, and they get out. Some have a lysogenic and lytic cycle where the phage infect and then integrate into the chromosome, so they become part of the chromosome. And then when they pop back out and go lytic, that's when they can grab a piece of this bacterial chromosome with them, and transduction happens. And the last slide we have for bacteria is talking about virulence factors. So this is anything that increases pathogenicity or disease causing. And this is trying to just kind of summarize some of the things that we've talked about. So we've talked about LPS, an endotoxin. An endotoxin means it stays with the bacteria. That's, remember, in gram negative. We have things like O antigen, which inhibits phagocytes, and we'll talk about the immune system. We have flagella. We have capsules. There are things called cytotoxins, which affect the surrounding eukaryotic cells. Enterotoxins, and we also call these exotoxins because they are produced and secreted into the environment and effects. So lots of different ways that bacteria can cause disease or pathology when it gets into a human or animal or yeah, I guess that would be it. Oh, and we have one last, slide that just wants you to understand these terms: genomics, proteomics. You've probably heard of them, but remember, genomics is information about genes that an organism encodes. and proteomics is the proteins that are expressed. Right? So just like your cells, you have the same genomics, same genetic information in all your cells, but different cell types produce different proteins. So the proteomics, or the proteins that are expressed, are different. And so by studying genomics and proteomics, we can get a better understanding of how pathogens, not just bacteria, but viruses, protists, can cause disease. Okay, that's it for now. The next video will be on viruses. Thanks.